Hi everyone, my name is Rachel.
Hi, I'm Casey, and Rachel and I assessed the motor responses and oxidative stress of in honeybees that were exposed to either neonicotinoids or pyrethroid pesticides, which are bee friendly. So, honeybees are major pollinators, and they've recently been shown to be affected by colony collapse disorder or CCD, where worker bees just abandon the hive. And this can be caused by many things, including exposure to harmful pesticides. And a commonly used pesticide with known harmful effects on honeybees is imidacloprid, which is a neonicotinoid. And some previous research conducted by another student from Bloomsburg University suggested that honeybees were affected by imidacloprid at only 1/10 of the LD50. So Rachel and I wanted to investigate some less toxic alternatives to potentially decrease cases of CCD.
Okay, so the two pesticides that we looked at were acetamiprid, which is a neonicotinoid, and deltamethrin, which is a pyrethroid. And like Casey said, we wanted to compare the results of these two with that of imidacloprid.
And so our main focus was really just to identify the sublethal effects of these bee-friendly pesticides by looking at motor responses and cellular stress responses in the honeybees after they were exposed.
Okay, so we did this by collecting honeybees over here with a sucrose solution, and then we put them in these little tubes and transported them back to the lab to place them in the freezer to anesthetize them and slow their motor skills. And then we harnessed them in these modified centrifuge tubes so they couldn't get out, and we can give them the proper dosage of the pesticide. And then 4 hours post-treatment, we looked at their proboscis extension, their antenna, leg, and abdomen movement, and we gave them a score based on how well they were functioning. And then we looked at superoxide dismutase, which is an antioxidant enzyme that catalyzes the breakdown of the superoxide anion, and that is a marker of cellular stress in the honeybees. So our results for the motor responses, we saw a significant impairment at the highest dosage that they were given, which was 1/5 of the LD50. And for deltamethrin, we didn't see any significant impairment at any dosage.
And for the SOD activity, you can see here that really for both acetamiprid and deltamethrin, between the control and treatment groups, there really wasn't a significant difference. So this may suggest that, at least in terms of SOD activity, these two pesticides may actually be bee friendly. And so ultimately, we found that the motor responses were impaired by acetamiprid, but were not affected by deltamethrin, and that the SOD activity didn't really differ significantly between the treatment and control groups. So this suggests that acetamiprid and deltamethrin may be safer alternatives to more widely used harmful pesticides such as imidacloprid. So our future research will include investigating heat shock protein 70 or HSP70 for additional evidence of cellular stress and how it may affect the honeybee transcriptome.